Okay, so we have membrane dynamics coming up with this S1P4. Also, information for the next lecture exam. Just a note, cell structure and cell anatomy is on the exam, right? You're responsible for the material covered in the handout. That would be S1PX2. Also the basic chemistry, which is S1PX1. So don't forget those, that you're responsible for them, and you will see them on the exam. We talk about membranes. Just a quick note about the terms. There's a number of different types of membranes. Like as an example, there's the pericardial membrane, right, that surrounds the heart. There's synovial membranes, and that was a serous membrane and things like that. But what we're talking about is the cell membrane, discussing the phospholipid bilayer. Okay, so in this case, we'll look at not the properties of the cell membrane, that's in the cell portion that I just mentioned, but we'll sort of look at what it takes to go across this membrane and move things from one side of a cell to the other. So the cell membranes in general perform various functions. The cell membrane provides physical isolation. It keeps the inside of the cell isolated from the outside of the cell. It allows us to regulate the exchange with the environment, so we can allow things to come in or leave. It provides communication between the cell and its environment. It provides structural support for the cell, and helps to separate charge and keep the charge separations very important in terms of how we function. There's a bunch of ways to look at cell membrane composition, but I think the easiest, and while not exactly accurate, is to remember there's two types of lipids, proteins, and carbohydrates in the cell membrane. You have phospholipids, cholesterol are the lipid portions, integral and peripheral proteins are the protein portions, and then the carbohydrates are glycolipids and glycoproteins. So when we move from one side of a membrane to the other, oftentimes we use channels, and we'll talk a little more about channels a little later, but we'll give you a nice, easy way to kind of remember them. So channels are proteins, and they can come in a couple different types. One type is non-gated. Non-gated channels don't have a gate. They're not very common, although there are some in every cell, but it gives you an open passageway between the inside and outside of the cell. When things go through channels, they work by diffusion. We'll talk about that again in a little more detail. So non-gated channels don't give you a whole lot of control. We talked about the four basic themes of physiology in the previous section, and while those four things are very important, I think if I was making it, one of the things I would add to that is the ability to have control. And control is very important, and you'll see a couple times when we talk about function this semester where our body has perhaps not the fastest or the most convenient method to do things, but that method typically gives us more control. And so we don't see a whole lot of non-gated channels because things are always moving through, and we have a lack of control. Generally speaking, most of the channels are gated, meaning they have a single or double gate that allows us to control the entry and exit. For single gated, it's very simple. The gate is open and things move. Gate is closed, things don't. For double gated channel, they're a little more complicated, where obviously both gates have to be open in order for you to move things from one side of the membrane to the other. So it's sort of like, walking into a classroom in a building at school. Right? So you have to get into the building first, that's like the first gate, and then you have to be able to get in the classroom, that's the second. And if both doors aren't open, then you can't actually get into the classroom. Now, there are a number of things that allow us to open or close gates. And really what it's talked about is it says we change the channel configuration. And things that can change the channel configuration would be chemical. So we have what are called chemically gated channels. We have voltage. Those are voltage gated channels, some unit of electricity basically. Mechanically gated channels. These mechanical channels open or close by mechanical changes like, pressure or touch or something. We have light gated channels. We have light gated channels all over the place in our body, actually not just our eyes, but in our foreheads and even our popliteal folds. So those are opened or closed with photons of light. We have time gated channels that open or close after a certain amount of time. We have temperature gated channels that open or close at a given temperature. Now, one of the things the cell membrane does for us is it helps us to separate the body fluid compartments. Let's take a quick look at them here. One of the things that we have scientifically is what I refer to as the magical, mythical, male medical model. So there's a whole bunch of M's. And what we kind of go with these standard values is a college-age male who weighs 150 pounds, which is equal to about 70 kilograms. And the problem with using this model is that obviously more than half the population is not male. In addition, many of the males are not college age, and in addition, even the college age males, very few of them actually weigh 70 kilograms. But this is the standard reference model that we have medically oftentimes. And sometimes, like on a TEAS test or a HESI or some other standardized exam, you might be asked what this is. So if we look at and use this 70 kilogram male, your total body water, the amount of water you have in your body, is about 60% of your body weight. It's a little more when you're an infant, and it's a little less when you're 70 years old, but it's pretty close to 60%. Okay, so if you take 70 kgs times 0.6, that would be 60%, you come out with 42 kgs. Now, the great thing about the metric system is 1 kg of water is 1 liter, right? And so if we have 42 kgs roughly of water, we have 42 liters. So our total body water is said to be 42 liters. We'll see later there's probably a better way to estimate that, but that's the magic number we're responsible for. Of that 42 liters, about 2/3 of it is inside the cell. If it's inside the cell, it's called intracellular fluid, or ICF, so intra means inside. 2/3 of 42 liters is 28 liters. And outside the cell, separated by the cell membrane, is the extracellular fluid. That's 1/3. So the only other place you can be if you're not inside a cell, you have to be outside the cell. That's ECF. That's about 14 liters. Now, of the extracellular fluid, there's a couple places where we can find that. This doesn't do a great job identifying them all. The plasma is about 3.5 liters. That's the liquid portion of your blood. The interstitial fluids, probably 9.5 to 10.5 liters. And there's another one not on here called the transcellular fluid. Okay, and I don't have it on the next slide. I thought I did. Transcellular fluid is the fluid that doesn't fit any of the categories I discussed so far. So it's not inside a cell. It's not interstitial fluid where it bathes the cell, but it's somewhere else. So examples of that would be the intraocular fluid. So many of you probably your anatomy class did a dissection where you opened up an eyeball, and the goo that split out, the vitreous and aqueous humors, those are considered to be, generally speaking, transcellular fluid. Your synovial fluid is transcellular fluid. So usually that's just, considered to be a small amount, less than a liter. So extracellular fluid is made up of 3 components. the plasma, which is a liquid portion of blood, the interstitial fluid that bathes the cells, and then anything that doesn't fit those three categories is called transcellular fluid. When I mean three categories, I'm talking about the intracellular fluid, the interstitial fluid, or the plasma. Now, the cell membrane is selectively permeable. And actually, it's better in some ways, again, because of control, that things can't pass directly through it. When we look at a cell membrane, right, this is just showing the phospholipids here. And the phospholipids line up in these, kind of unique arrangement that sometimes is referred to as the head, tail, tail, head arrangements. Now, phospholipids, right, are really just when you take a triglyceride. Remember, triglyceride was a glycerol molecule with 3 fatty acids. Remove one of the fatty acids and put on a phosphate in its place. Okay, so what happens is when you do that, you make this molecule polar on one end. The glycerol is a sugar alcohol, which is polar. The phosphate is definitely polar because it has that -2 charge. And the fatty acid, there's still 2 left. Remember, those have methyl groups, and the methyl groups are the nonpolar group. So the fatty acids tend to be nonpolar. When these aggregate in a solution and form a cell, what happens is the fatty acids stick together. So we have these 2 acid tails together, fatty acid tails together, and the heads, the polar region, right, stick out towards the outside. And so again, it's called the head, tail, tail, head arrangement. Because the heads are made up of glycerol and phosphate, they are polar. Polar things are often said to be hydrophilic. Hydro, water, philic, loving. So they like to be around water. Again, it's called the head. Sometimes they called, say, to be lipophobic. Okay. Now, the tails are nonpolar. So this pink area is nonpolar. There are fatty acid tails. Those are said to be hydrophobic, water-fearing. And then we do the exact same thing on the opposite side, but we go tail, head. Ben Franklin actually did this experiment back in the 1700s, and what he saw is if you put a oil solution on a pond, he was able to look at the volume of oil he put on the pond versus the surface area of the pond and calculate exactly what was happening. And even though he really couldn't see it, he surmised that what happened is the polar heads stayed in the water and the fatty acid tails literally stuck straight out of the water to get as far away from water as possible. So when these aggregate, they aggregate in this manner. Now, one of the things that we can say about then the phospholipids, you can also say it about a triglyceride as well, is that they're amphipathic. And an amphipathic molecule has polar and nonpolar components. And amphipathic molecules are going to be important in lab later on, and we'll have some other importance during the semester. But amphipathic, it's sort of like amphibian, right? An amphibian is an organism that lives in both land and water. So amphipathic means it has both polar and nonpolar ends. So when these aggregate to form a cell membrane, what happens is this bilayer right here, right? So there's 2 phospholipids, the top and the bottom, so it's called a bilayer. The inside forms this nonpolar hydrophobic portion. And in order to be able to move from one side of the cell to the other directly through the phospholipid bilayer, you need to have 2 properties, okay? Number 1, you need to be soluble in the bilayer. Okay, so you need to be what they call lipophilic, or sometimes lipophobic. lipid soluble, right? So you need that. In addition, you need to be able to physically wiggle through these, so you need to be relatively small. Okay? So in order to pass directly through the membrane, you need to be small and nonpolar. Examples of that would be gases like oxygen, carbon dioxide, glycerol, and other types of alcohols. Number of lipids like steroid hormones, things like that can go directly across. And interestingly, water can go across directly, which is not nonpolar. So it's small, but it's polar. Recent studies have shown that water doesn't move as much as we used to think it does by itself across, but it still does go across. So it is somewhat unique. If they don't fit that category, substances are then typically impermeable. And impermeable means the substance cannot penetrate the membrane directly. So large molecules, lipophobic molecules, right, cannot go directly across. So examples of things that can't go directly across: sodium, potassium, chloride, calcium, glucose, all examples of chemicals that can't go across. And you can't get much smaller than sodium in terms of a size of a particle, right? But it's an ion, and ions are very charged, and it can't go across because of that. Glucose doesn't have a net charge like any of the ions we see here before it, but it does have a charge distribution, which makes it polar. And also it's pretty big. It's got a molecular weight of 180. So what determines how things get across? Sorry, didn't want to do that. It's influenced by a couple things. Particle size. Smaller molecules go through faster. Particle solubility. The more soluble you are in the membrane, the faster you go through. Particle concentration. The higher the concentration difference from one side of the membrane to the other, the faster a molecule goes through. And finally, membrane content. The more cholesterol you have in a membrane, that's going to affect how fast things go through. And basically, it has to go through this phospholipid portion. And so the less phospholipids you have, the harder it is for things to get through, because it's harder to find that phospholipid portion. So all of those factors are going to influence the permeability of a molecule. And so the cell membrane is said to be selectively or semi-permeable. Now there's two basic ways to move across a membrane. One is what we call passive transport, and the other is called active transport. For passive transport, there's no energy input required. What we're doing is we're relying on the energy already in the system. Okay, and it's actually the energy from the kinetic movement of the molecules. Okay, so passive transport doesn't require any energy input. And particles move down a gradient from a high to a low something. Oftentimes it's concentration. In physiology, there are some things that move by pressure, but we're going from high to low. We can also move things across a membrane by what we call active transport. Active transport, by definition, requires some input of ATP at some point in time. We have to expend biological energy to move things. And when we do that, invariably, at least one particle, one substance is moving against a gradient. Something is going from a low to high something. And usually it's a low to high concentration or a low to high pressure. And well, it doesn't work exactly. Not a bad way to envision active and passive transport is if you were vacationing on a river and you hopped in a boat and floated down the river with the current. That would be passive transport. You're using the energy already available to you in the river, and it moves you down. Okay, so you're going from high to low. But if you want to go back to where you started, now you have to go against the current. You have to put in energy, whether it's turn on a motor or row or whatever it is, to move the boat back up river against the current to where you started. So that's like an active transport. mechanism. So when we look at passive transport, we're going to study 5 different passive transport mechanisms in the body. There are more. We're not going to talk about all of them. We're just going to talk about these 5. All right? These are the 5 you're responsible for. Simple diffusion, which isn't even moving across a membrane, to be honest. Simple diffusion could happen in, a cup of water, right? So if you take a cup of water and just water, and put a drop of food coloring in and let it sit there, eventually the food coloring will go from just that little spot where you put it in to being distributed throughout the water. That would be simple diffusion. Okay? Diffusion across a membrane, we're going to talk about that. That's going directly across. We sort of mentioned the properties you need to do that, small and nonpolar. Diffusion through a channel, we've talked about that a little bit. That's what it's showing here in this picture. Right? Here's a channel. Moving things from one side to the other. Facilitated diffusion, which is using a transporter to go. And then osmosis, which is a special case of diffusion with water. So those are the 5 we're going to talk about. So as an example, solvent drag and bulk flow or other types of passive transport mechanisms, we're not going to talk about them though. So let's start with simple diffusion. Simple diffusion, and this is going to be true of most of our definitions with passive transport, will be net movement of like particles. It's not just ions, it's like particles, right? From an area of high to low concentration. And again, a great example of that would be something like, here's a purple dye, and each one of these little circles, right, represents a dye molecule. And we put it into the solution initially, right? It's all concentrated to the left side here. And after a period of time, it's distributed equally. Okay? Now, one of the things to remember is that once we have it distributed equally, the particles don't stop moving, right? What they do is they reach what's called dynamic equilibrium. And for every purple particle that's going to the right, there's one going to the left, and there's one going up, and one going down, one going backward, one going forward, and so on. So the concentration is equalized because we have a dynamic equilibrium, right? But since it's dynamic, the particles never stop moving, at least practically for us. Yeah, we could talk about, absolute zero and things like that, but we won't worry about it physiologically. Because at absolute zero, kinetic energy stops, and all motion stops, and really, for the most part, all function stops then, at least as far as we know. So things that can influence simple diffusion, right? So here's an example of putting sugar cubes into coffee. All right, now, if this coffee was warm, right, what's going to happen to the sugar cubes? They're going to dissolve faster. If this coffee was ice cold, the sugar cubes wouldn't dissolve as fast. So temperature increases the rate of diffusion. And the reason why is pretty simple. Okay? Diffusion relies on the kinetic energy of the molecules. And the higher the temperature, the greater kinetic energy we have. The energy input itself. So if you don't want to sit there and wait for those sugar cubes to dissolve and eventually, move throughout your entire cup of coffee, you can stir it. So we can increase the energy input by stirring, which would also increase the speed at which the diffusion took place, so we could equally diffuse sugar throughout the solution so that every sip tasted the same. The other thing is molecular size. The larger the molecule, the slower it moves theoretically, so small molecules will increase the rate of diffusion compared to large molecules. The other 4 things we're going to talk about in terms of passive transport for this section involve diffusion across a membrane. And so you can kind of see it here. So this is not right. And, one of the problems with science is that oftentimes the good artists aren't the good anatomists or physiologists. And in many, many cases, we have models or figures that are not correct. Okay? So as an example, this is talking about simple diffusion. All right? And really, it identifies these molecules as transported, yet this molecule is going directly across the membrane. So it is not technically transported. It's moving all by itself, right? This is a transported molecule here, but technically it goes through a channel, and then we have these carriers, and we'll talk about uniports, symports, and antiports when we get to facilitated diffusion. Might as well do it now because it's on this graph. So when we see the word port, it's referring to transporter. And this is true of not just a facilitated diffusion, but it's true of other things like active transport, things like that. So anytime we have a transporter that carries a molecule from one side of the membrane to the other, we can give it some general names. If the transporter transports one thing in one direction, it's called a uniport. Much like if someone rode into the classroom with a bicycle with one wheel, you'd say, oh, that's a unicycle, right? So uni means one. So a uniport will move across the membrane, right? One direction. It doesn't have to be just out to in. It can be in to out. It doesn't matter, but one molecule, one direction. A symport, right? Sim means together. So in symport is when we move two or more molecules in the same direction, okay? And again, oftentimes it's in the cell, but theoretically it could be two molecules out. An antiport, anti means against, right? So an antiport moves at least one molecule in while moving at least one molecule out, all right? So those are kind of the terms we see for these different transporters. When we move more than one molecule in any way, that's technically called coupled transport, right? Because there's more than one, so it's a couple, right? And it could be 3 or 4 or whatever, not just 2, but anything more than one is called coupled transport. Now, one of the things that we want to look at is moving molecules directly across the membrane. So let's go back to this one right here, okay? So here's my molecule, this little circle here, and it's moving directly across the phospholipid bilayer. These are supposed to represent the heads of the phospholipid. The red are the tails. It's our lipid bilayer. It goes head, tail, tail, head. And so what properties must this molecule have to move from one side to the other? Give me a second. It must be A, small, and B, nonpolar or lipid soluble. Not always exactly the same, but for us, close enough. Okay, so it has to be soluble in the lipid bilayer here, right, in the fatty acid tails. It has to be small enough to wiggle through. Okay. In addition, because we're talking about diffusion, right, all of these molecules in this case, right, we won't worry about the antiport necessarily, but all these molecules in this case, we have this one moving from this side of the cell, the top, down to the bottom. So where is the concentration the highest for this purple circle molecule? Well, since we know we're moving from the top to the bottom, and it's diffusion, it means we have to go from a high to low concentration. So the circle has to be a higher concentration on the top in the yellow-brown than on the bottom in the blue. Okay. So how do we go directly across the membrane? Well, let's kind of take a look at some of the factors that matter. So here's a situation where we have let's say two containers. They're separated by a membrane. That's the dotted line here. Okay. On the left side in A, where the red is, notice all the particles on the top, no particles on the bottom. And in B, there's much fewer particles on the top, but still no particles on the bottom. And then we go ahead and we let this do its thing. Where is the flux the greatest? What flux refers to is the appearance of the molecule on the other side of the membrane. Okay? And hopefully you can all kind of think in your head and see that A will have a faster, what we call net flux technically, than B. that more molecules will come across the membrane on A than it will B. And it's really simple. In order to go across the membrane, what do you have to do? Well, you have to be moving in the direction towards the membrane to strike it. So with only 6 molecules, you're less likely to strike the membrane than if you have, 15 or 20. Okay? So the higher the concentration, the greater the net flux. Now, this situation, we have A and B. What we did is we put the exact same number of particles in both, right? Because we know that that makes a difference. And in A, here's my membrane here, these little dotted, but right here at the beginning and right here at the end is plastic. And the molecules cannot go across the plastic. They're not soluble in it. While in B, we have this membrane go all the way across. Okay, so which one's going to have the greatest flux? Well, we know that the number of molecules doesn't make a difference because they're the same, so the concentration's the same. But what is different is this plastic portion, right? And anytime a molecule hits the plastic portion, it can't get through and it's going to bounce back out. In this one, if it hit the relatively the same place on B than it would A, because it's membrane, not plastic, it would go through, right? So it's sort of like if I said, okay, I'm going to draw a target on the board, and if you hit the target, you get an A in the class. If I drew a little tiny target, it would be harder to hit than if I drew a big, huge target, right? So since a number of molecules are going to hit the plastic and bounce back, B is going to have a much faster flux than A. So what we call the difference between A and B is B has a increased surface area. And the bigger the surface area, the increase in the net flux, the faster the diffusion goes. Now we're going to start with kind of the same parameters in A and B. So A and B, same number of molecules. A and B, same size membrane. But this time in A, that's why we have it, the dotted line in kind of orange, we're going to make the resistance of membrane A greater than membrane B. And so what happens is, yeah, the molecules are going to hit the membrane at the same rate because they have the same concentration, right? They have the same membrane size, they hit at the same rate. But since there's greater resistance in A, what's going to happen is that's going to slow the molecules as they move through the membrane. And since the molecules are slow through the membrane, their appearance on the other side is going to be slower. And so in this case, the flux in B will be greater than the flux in A because the molecules move through the membrane faster because there's less resistance. Okay? What does resistance depend on? Well, a number of different things. The membrane composition and the particle size and solubility. We'll do an experiment in lab that sort of looks at some of those properties. But the bottom line is they're not going to be the same. So membrane resistance depends on the membrane composition and the particle size and solubility. And as we increase the membrane resistance, that decreases the net flux, right? So on A, we've increased the resistance, and that's going to cause less particles to appear at the bottom. So we're decreasing the flux. All right, the last one is the idea of membrane thickness. So what we've done is, okay, we know we have to have the same molecules, and we know we have to have the same surface area, and we know we have to have the same resistance. So what we did in A is we made these membranes thicker, right? And again, as you can probably guess at this point in time, as the molecules hit the membrane, they're going to travel slower. And since the membrane is thicker, it's going to take them longer to get through and appear the other side. So the flux in B is going to be greater than the flux in A, again, because the molecules don't spend as much time in the membrane because it's not as thick. So just like we had in the last one, membrane thickness, if we increase the membrane thickness, that decreases the net flux. Okay? So kind of to summarize this, the net flux is directly proportional to the concentration gradient and the membrane surface area. Right? We saw if we increase the concentration gradient, the difference between one side and the other, flux went up, and the membrane surface area. If we've increased the membrane surface area, flux goes up. But it's inversely proportional, the net flux, inversely proportional to membrane resistance. If resistance goes up, flux goes down, and membrane thickness. If thickness goes up, flux goes down. So what we can do is we can set this up into an equation. And the equation is pretty simple in terms of how it works. We can say the net flux, which is sometimes called J, right? We'll just call it the net flux, equals the things that are directly proportional divided by the things that are inversely proportional. So net flux equals concentration gradient times surface area divided by membrane resistance times membrane thickness. Okay? So if the concentration goes up, net flux goes up. Surface area goes up mathematically, net flux goes up. Membrane resistance goes up, flux will go down because it's inverse proportional. Membrane thickness goes up, flux will go down. Okay? Then you have to remember, though, it only applies to direct diffusion of particles across a membrane. It only applies to small and nonpolar molecules. Well, the first person to kind of put this together in a mathematical sense was a scientist who did a lot of physical, functional things in the human body, Fick, and so it's called Fick's law. So Fick's law of diffusion, right, summarizes this, and this is the equation. So if I ask you to use Fick's law to figure something out, you probably want to figure it out that way. And we'll see some examples of Fick's law and how you can apply it later. Well, if we don't go directly across, then we need to go some other way. And another common way to go across is diffusion through a channel. And a channel is sort of like an aqueous pore for diffusion of lipophilic properties. So molecules that just can't get across the membrane will use channels oftentimes. And channels are a protein. They're integral proteins because they expand across the entire membrane. And a couple other things to be about channels. They're very fast. Okay, they work by passive transport. As we said before, they can be gated or non-gated. They tend to be limited to smaller things like water and ions. As far as we know, there are no glucose channels in terms of a cell membrane. Now, glucose can get in and out of a cell for various reasons, but there's no channels for them. And the flux will depend on a number of different factors. The concentration difference from the particles, the bigger the concentration difference, the faster it moves. The number of channels available and open, the more channels you have, the more you can move, right? And one of the things, and it kind of won't make sense a lot now, but it may later, hopefully, is channels cannot be saturated, meaning that whenever we increase the concentration of a substance, the flux in the cell is going to go up. We cannot concentrate channels. Yeah, if you pulled a channel out and studied it, what we call in vitro, right? In a laboratory situation, you made like 1,000,000 times difference in a chemical, then you could theoretically saturate a channel where if you increase the concentration, you wouldn't increase the flux. But physiologically, we can't do that naturally because our concentrations have to maintain in pretty steady amounts. And if something changes too much, we'll die. So yeah, you can't, for our purposes, you cannot saturate a channel physiologically. Now, the review of channels we talked about before, right? Channels can be always open. Right? And sometimes we have like an example, like a potassium leak channel in our cells. And it just means that the movement of the particle depends on what the concentration difference is. So in humans, if we look at a like a potassium leak channel, potassium actually has a higher concentration inside the cell than outside the cell. And what happens is potassium is always leaking down its gradient, high inside, low outside, and is always leaking out of the cell. Okay, we'll see why that's important a little later, but it helps the inside to maintain a negative charge, and that's because potassium is a positive charge, and if you're always removing potassium from the inside, you have less positive charges. Then we said that you can have gated channels, right? And gated channels can either be open, where things move, or closed. This is a single gated channel. Here's one on the bottom that's a little easier to kind of represent this little line here. And you could put a double gated channel in where you'd put a second line here in terms of that. And things that typically change the gate configuration in the human body, again, are chemicals, voltage, mechanical stress, light, time, and temperature. Now, when we look at other channels, it does rely on diffusion, so it's simple diffusion, and things are just going to diffuse through the channel. Channels are specific, like, so as an example, this potassium leak channel won't move anything but potassium. We have channels for water, we have channels for all sorts of things now that we've discovered. Another way to get across is not through a channel, but actually with a physical transporter. And basically what happens in a transporter is the molecule is grabbed on by the transporter, and the transporter physically moves it from one side to the other. They're significantly slower than channels, and oftentimes, 10 times slower, and sometimes hundreds of, even thousands of times slower. But we're always moving, again, from a high concentration to a low concentration because it is a form of passive transport. So all facilitated diffusion means helper diffusion. And so our definition for that is net movement of like particles using a protein transporter from an area of high to low concentration. Right? Transporters are very specific, right? And they bind specific substrates that we call ligands. People say ligand, but ligands, right? And for our purposes, a ligand, a nice, easy, simple definition of a ligand, is anything that binds to a receptor. Right? And what happens is the ligand binds, it changes the channel conformation, and it transports it across from one side to the other. The reason why this is slower than a channel is it never creates a continuous passage from inside to outside. Right? So if you look at this middle figure, there's not a continuous passage. In a channel, there is a continuous passage, which allows the molecules to move through faster. So facilitated diffusion is just helper diffusion. It's a protein that enables a molecule to move from one side of the membrane to the other down its concentration gradient. Okay, so it's called facilitated diffusion. All protein transporters that have a receptor exhibit 3 different properties. And these properties are very important. Learn them now because we're going to see them again and again and again. They exhibit what we call specificity, right? And that means they bind specific ligands or occasionally classes of ligands, right? So ligand, again, is any molecule that binds to a receptor. As an example, there are some transporters in our body that only bind glucose, no matter what, just glucose, right? Really specific. There are other transporters in our body that will bind any 6-carbon monosaccharide. So specificity is still specific. So they have the selectivity in terms of the binding site. It has to do with its 3-dimensional shape. So that's specificity. Competition is ligands compete for binding sites. All right, and then sometimes related substances, depending on their concentration and affinity, may be able to outcompete other things to move across. As an example, amino acids in our diet are absorbed through the gut by primarily this process. And what happens is that if you eat more of, amino acid like lysine for some reason, then you're going to absorb more lysine because it's going to compete better for the facilitated diffusion binding site, which then moves it into the blood from the gut, right? And there's two things that determine competition. One I just mentioned, concentration. The higher the concentration, the more likely a ligand will meet the receptor, which means more likely that reaction will happen. But you can make up for concentration, if you have a lack of concentration, with what's called affinity. Affinity is just a nice scientific word to say attraction. So if there's a greater attraction, a greater affinity of one molecule to another, it will bind more preferentially at that point in time. Okay? So the two things that determine competition, concentration, the higher concentration, the greater the binding and the greater the diffusion then. And affinity, the higher the attraction, the greater the binding and the greater the diffusion. In addition, for competition, you can have what's called competitive inhibition, where something blocks something from binding and inhibits it. We'll see that more a little bit in, we'll talk about enzymes, okay? The last point, which is very important, because this has come up a couple times, is the idea of saturation. Saturation is the rate of what we call transport maximum. As long as you increase the amount of ligands, right, the transport rate will increase until all of the transporters, all of, in this case, facilitated diffusion transporters, are full. Once they're all full, and we've reached what they call 100% saturation, we can't go any faster because everything is full right now. And so it depends on the substrate and the transporter concentration to do that. And, as an example, one of the things we have is we have transporters in our kidneys to remove glucose from the urine. And somebody who's a diabetic, one of the hallmark symptoms they have for uncontrolled diabetes is they have what's called glycosuria, glycose, sugar, urea, urine. So when someone has sugar in their urine, it's because they've saturated the transporters in their kidney and they can't remove all the glucose, and it just ends up staying in the urine and you pee it out. So the three properties we see for protein transporters: saturation, competition, and specificity. Now, one of the things that cells do is, they want to try to avoid equilibrium. And, glucose is very important for us to get into the cell, and this is a really good example of how this works. Okay, so we have this high glucose outside the cell, these little pink, looks like minus signs, I guess they're purple, right? And here's the cell. And so what we do is we have facilitated diffusion that takes the glucose and moves it into the cell. Now, if we move enough glucose into the cell, right, we decrease the concentration outside, increase the concentration inside, and they reach equilibrium. When they reach equilibrium, we'll no longer have the passive movement of glucose into the cell. That creates a couple problems, because one, we don't want glucose to be that high on the outside of the cell anyway. And two, if we don't need it outside the cell, we need a place to store it, which is inside the cell. All right, so what we do is, instead of having glucose just hang around, we turn glucose into glycogen. Or we could use it for energy through glycolysis. And once we have glycogen stored, then it's no longer glucose, and we can keep moving glucose down its concentration gradient because we're not accumulating in the cell, because we're either using it for energy or we're storing it as glycogen. And not all of our glucose works by facilitated diffusion, but a lot of it is. And it's nice to not have to expend any energy to put glucose into the cell. So cells avoid reaching equilibrium oftentimes by metabolizing substrates or changing their identities. The next and last passive transport mechanism is osmosis. But since this video has been fairly long and we probably got, one that's almost as long left, let's stop here and then pick up with osmosis on the next video in this series.